So yeah, we're just going to start with a quick review of what we did last time, just so we're clear on what nerve tissue is, right? So this was about neurons and how they get myelinated, and that was by neuroglia, the Schwann cell, and then of course the neuroglia. And so last month we started with just a review of way back, one of the first PowerPoints we had, the introduction one, and then we went into this one where we just had a quick review of the neuron. So as we know, the neuron is made up of three parts: the cell body, the dendrites, and the axons. We looked at this structure of a multipolar neuron, which is all of your motor neurons, your lower motor neurons. So this is what the neurons that go to all of your skeletal muscles look like. Okay, so that cell body in this type of neuron is up in the anterior gray horn of your spinal cord. And then the long axon is inside those nerves we learned about when we took spinal nerves. And so inside the spinal nerve are all these tiny little threads, which are the axons of all the multipolar neurons. As we saw, the axon of a neuron is the part that will probably be myelinated. Okay? In some places, maybe in the CNS, you might see dendrites myelinated, but mostly it is our axons that are myelinated. And in the case of this multipolar neuron, the axon is myelinated. It's in the peripheral nervous system, so the neuroglial cell that does that is the Schwann cell, or the neurolemmacyte. Okay? So the neurolemmacyte does the myelin on the axons in the peripheral nervous system. And as you can see, it's just this fatty extra coating of phospholipids. Okay, so the cell itself, the neurolemmacyte or the Schwann cell, is a typical cell, but this cell wraps itself around the axon. You can see its nucleus in the outside in the neurolemma, okay, which is the outside of the neurolemmacyte. Okay, and then on the inside, it just makes more of those phospholipids that we saw in the phospholipid bilayer when we drew the cell way back in physiology. It makes extra of that to actually insulate the axon because that's a fatty coating. Okay, so it's in these nodes of Ranvier, the neurofibril nodes, in between the coating of the myelin and the neurolemmacyte, where we have collections of sodium channels. Okay, so that's what we're going to be talking about today, is these sodium channels, and they're actually more dense at these neurofibril nodes, or the node of Ranvier. Okay, so we're going to see these voltage-gated sodium channels are more dense at the neurofibril node. Okay, so when the impulse happens in this typical neuron, it starts in this initial segment here at where the axon hillock, which is where the cell body starts to taper here. And then in there is the initial segment. And in the initial segment is an area called the trigger zone. And the trigger zone is going to have the first voltage-gated channel. And what that means is that every sodium channel all along these neurofibril nodes is actually activated by a change in voltage. And as we know, in the human body, we can't have just electrical signals. They have to be electrochemical. Can anybody tell me what chemicals that we would use that would be electrical? What did we learn about way back in chapter 2? Potassium would be one, calcium would be another. So what are we looking at? Ions, right? So cations being positively charged and anions being negatively charged. And so they give us these electrochemical signals that happen, which we would call a nerve impulse or an action potential. Okay, so it's at this initial segment in here where you're going to have ones that are voltage gated, the start of them. And the way voltage gated work is, you may have been reading, or hopefully you've been reading, it's an all or nothing kind of thing. Once the first gate pops open that's voltage gated, all the rest of the voltage gated channels all the way down the axon will pop open because of the positively charged sodium that crosses the membrane. Okay, so you pop open a door here at the start, okay, in this initial segment, and that comes from these dendrites and graded potentials, and we'll be looking at those, and those are usually caused by neurotransmitters. And you get these slight graded potentials, and if they build up enough, they pop open the first voltage gated channel. Exactly, Angela, like dominoes, okay? So once the first one pops open, because all the rest down here pop open because of a change in polarity or electricity, when that happens, 20,000 sodium, which all carry a positive charge, dump into that first gate. And then that changes the voltage all the way along. All these gates pop open and it pops along. Okay, so we see that that whole process happens along the axon. It will travel down to the axon terminals, and when it gets to the synaptic end bulb, there's going to be more voltage gated channels, but this time somebody had mentioned calcium. Okay, so at the very end in the synaptic end bulb is a calcium voltage channel, and it pops open the same as all the sodium voltage gated channels. The calcium channel pops open. Calcium goes into this little swelling on the end, and that causes the neurotransmitters that are in the vesicles to be released. Okay, so you have sodium, sodium, sodium rushing in, and that's what your nerve impulse is. That travels to the very end to the synaptic end bulb. That opens up a calcium voltage gated channel. That causes the vesicles to go to the very end of the end bulb and dump out the neurotransmitter into the synaptic cleft. And then that's making your muscle contract right now. Okay, so that's why we need to understand the structure of a neuron, because you get these graded potentials at the dendrite, and then that's going to hopefully build up enough to cause the voltage gated channel to open up in the initial segment, and then all the way down our axon, which we've been learning about for a long time now, you get this impulse. Okay, that pops open all these channels all the way to the end where there's a calcium channel. And then that will cause the vesicles filled with neurotransmitter, which were made up here in the cell body, in the nasal bodies. You might remember those were the ribosomes of a neuron. And that's going to cause those all travel down the microtubules all the way down to be stored in the synaptic end bulb. They'll finally be released to cause a muscle to contract, a gland to secrete, or another nerve to have an impulse. Okay, so we learned about the cell body, the dendrites, and the axons, where we have these nerve impulses, and of course the trigger zone, where we have the first of the voltage gated channels. Okay, so we started talking about myelin last time as well. In the peripheral nervous system, the Schwann cell, or the neurolemmacyte, produces the myelin, and there we can see the Schwann cells. If we just go to the picture, okay, the Schwann cell wraps itself around the axon, and then internally, closest to the axon, it starts creating like rolls and rolls and rolls of phospholipids. Okay, and then that electrically insulates the axon in that spot, leaving exposed wire or axon only at the node of Ranvier or neurofibril node. Okay, and that's where you're going to have the collections of sodium voltage gated channels. Okay, they don't need to be on the other part of the neuron that's insulated because it's insulated. Okay, so they're just going to be collected in the neurofibril node. We can see that in the C fiber, which we know is unmyelinated, it would look more like this. The neurolemmacyte, or the Schwann cell, is still going to provide some protection to the cell, but it doesn't myelinate it. It doesn't do that wrapping underneath. So it sort of protects a bunch at once, and you can see it doesn't create that insulator, the phospholipid, all wrapping around it. We then moved on to talk about the neuroglia of the central nervous system. So nerve tissue is neurons and neuroglia. We know that the neuroglia outnumber the neurons 50 to 1. So there's way more of the neuroglia than the neurons. In other words, the neurons are the rock stars, and the neuroglia are everybody that makes sure the rock star can get on stage. Okay, so you've got all the people that book the facilities, you've got the managers, and you've got the roadies, and you've got the truck drivers, and you've got the bus drivers, and you've got the people that cook the food, and you got all of those are the 50 to 1. Okay, so neuroglia can multiply and divide. They make up most of your nervous system. And the ones in the picture on page 412 in your text, in the picture are your central nervous system neuroglia. Okay, so in the picture. In that same chart on 412, you also have listed the 2 that are in the peripheral nervous system. So there's 4 in the central nervous system, 2 in the peripheral nervous system. So inside your central nervous system, which you know is your brain and your spinal cord, you have the astrocyte. Okay, so there's different types of astrocytes. They don't get into it in your book, but there are different types. Okay, there are the microglial cells. Okay, the microglial cells are the ones that are like the phagocytes of the central nervous system. So they're like a fixed macrophage. They're right within the tissue, and they're going to take up, you know, any cells that have been damaged or died or anything like that. Any waste products that get into the nerve tissue, they're there to be phagocytic, so just clean it up, just like macrophages anywhere. Okay, then we had the oligodendrocyte. Looks like an octopus because it has all these different arms, and it produces the myelin in your central nervous system. So you can see it just reaches out, and it can myelinate up to about 15 neurons in the central nervous system. This is the myelin in your central nervous system that is affected when people have multiple sclerosis. Okay, so it's in the central nervous system that is the problem with that. So the myelin produced by the oligodendrocyte. And as you can see, it will myelinate quite a few neurons at the same time. And then last but not least is the ependymal cell. It lines the ventricles or the spaces in your brain. Okay, and there will be blood vessels and coming to it, and the blood will be filtered through the ependymal cell. The Schwann cell would be like the oligodendrocyte, yes. Yeah, they both produce myelin, but they, as you can see, they produce it quite differently. It can take up to 500 Schwann cells to myelinate one axon, whereas we can see one oligodendrocyte can myelinate up to 15 axons. So it's quite different myelin, right? And it's the myelin by the Schwann cell that allows cells to be able to repair in the peripheral nervous system, because you have all these individual cells doing the wrapping. So if you were to cut this nerve in a spot, if the neural lemmacyte was okay, it would be able to help that axon repair itself underneath. Whereas if you look at the myelin in the central nervous system, you just got one cell trying to myelinate a whole bunch of axons. It's not the only reason that there can't be repair, but it's a very big one. Okay? Okay, so in the central nervous system, it's very difficult for neurons to repair after they've been damaged. They'd rather rewire. Okay, so you've heard about neuroplasticity, so the brain can kind of rewire itself. So that's more than likely what will happen rather than repair of a damaged neuron. You have billions of them in the central nervous system, so the microglial cell will more than likely just eat it, the damaged one, rather than it try to repair. You're welcome. Okay, so the ependymal cells, they do the filtering of your blood, so that it creates on the other side where you can see all these cilia, okay, on the other side, so in the ventricles of your brain, the central canal of your spinal cord, and in the subarachnoid space, you have ependymal cells that are filtering the blood and producing cerebrospinal fluid to filter and go through those spots. And then we know that that is all over all of the nerve tissue. So you filter out all the crap that's in your blood and produce cerebrospinal fluid so that your cells can get what they need within the central nervous system without the risk of being in direct contact with the blood. Okay, so again, we can see the astrocytes have a bunch of functions. They're one of the busiest cells within the central nervous system. They can produce something called a gliotransmitter. So they don't really produce a neurotransmitter because that would be from a neuron. They're a glial cell, so they produce a gliotransmitter. And they actually are released wherever there is a synapse or a connection between two neurons. And what the astrocyte gliotransmitters can do is boost it or try to negate it. So two neurons are trying to have an impulse go between them. The astrocyte can say like, go, 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 and give it extra stuff to boost that and make sure it happens, or give it something so it doesn't. Okay. What it can also do is maintain tight junctions in your blood vessels. Okay, you don't want your dirty blood in contact with your blood vessels, so it's called the blood-brain barrier. And what happens is you actually have a thicker basement membrane in the capillaries of your central nervous system, especially your brain. And so it's a thicker basement membrane, and then, you know, normally when you have inflammation anywhere in the body, we know we have vasodilation and then increased permeability. Increased permeability means the cells have more space between them so that you can see that the macrophages and stuff can get into the tissue to heal it. That doesn't happen in your brain because of the blood-brain barrier. No increased permeability. You're going to have tight junctions maintained between the cells so that extra stuff can't get into your brain. And so the astrocytes actually help maintain the tight junctions so that all the stuff in your blood can't get through. So only the stuff can get through is what normally can go through a cell membrane. So that would be oxygen, carbon dioxide, alcohols, and fats. Okay, they can go directly through. Okay, other than that, you would need your cerebrospinal fluid, which we saw the ependymal cells creating, to be able to get glucose, etc. Okay. The microglial cells, as we saw, were the macrophages, the fixed macrophages within your central nervous system. So anything that might leak through, they're there to be able to clear away any dead cells, etc. We saw the oligodendrocyte, which was the octopus, creating myelin for up to 15 different axons within the central nervous system. And we learned again about the ependymal cells. So here you can see the blood coming to the area. You can see a term here called the choroid plexus. We don't really take about that until we take the brain, but all that is where the blood comes to the ependymal cells and the filtering occurs. So that whole area is called the choroid plexus, and it's in the ventricles of your brain. But again, you don't really need to know that right now. Right now, what you need to know is the ependymal cell is the cell that filters your blood to make cerebrospinal fluid. Okay? And then because of its cilia on the one side, it actually helps it circulate. So the cilia, just like mucus in your throat, the cilia move that. So does the cilia on the ependymal cells help circulate your cerebrospinal fluid. In the peripheral nervous system, again, they weren't in the picture on page 412. They're just listed there. The neurolemmacyte, or the Schwann cell, is what produces the myelin in the peripheral nervous system. Again, it can take up to 500 neurolemmacytes to produce the myelin for one axon. So those really long axons that have to go down to your big toe, you're going to have a lot of neurolemmacytes producing the myelin. And last but not least is the satellite cell, and you can see them in between the neurons here, the cell bodies, okay, of your unipolar neurons. If you remember, these satellite cells are in ganglia. So here we can see the dorsal root ganglia of your spinal nerve. The dorsal root ganglia we knew had the cell bodies of your unipolar neurons, so they're not alone in there. They actually have some help, the satellite cell. So it's there just to help making sure that the neurons, the rock stars, get what they need. So you can think of the astrocyte as the rock star's manager, okay? And then when they have to go to a concert, they have their personal assistant. And you can think of the satellite cells like their personal assistants when they're on site to be able to get whatever they need. That was mentioned to me the other day and I kind of liked it. So the satellite cells are kind of like, they take care of everything. You know, we'll get you the tequila you want and the girls you want and the boys you want, whatever you want. Okay. And those are in the ganglia. Now, it's not just dorsal root ganglia that we have. Those are the ones you learned about. But I think we've mentioned before, in your autonomic nervous system, you also have ganglia. Okay, so you're going to have lots of satellite cells in those ganglia. Does anybody remember what a ganglia is? What does that term mean? A collection. It's a swelling or a knot. Yeah, that's the actual definition. And then it is a collection of cell bodies. Correct. Okay. So you're going to have anything outside of the peripheral nervous system. But of course, then in the brain, they have a part they call the basal ganglia, which goes against that. But hey, I don't name this stuff. Anyway. Okay. So we learned when we took the spinal cord that the gray matter formed that butterfly or H shape in the center of the spinal cord, and the white matter was all around it. As we've learned, all that white matter is myelinated axons. Okay, so we learned when we took the spinal cord that the white matter is arranged with axons either bringing the information up to the brain or back from the brain, and then it's going to go out by its spinal nerve. So the white matter is formed into tracts, and they're just carrying information up and down the spinal cord. The gray matter is where connections are made, or where you'll find interneurons, the neurons in between two neurons. Okay, so gray matter contains those. When you look at the brain, the white matter is more in the middle and the gray matter is around the outsides. Okay? When you look at the brain, when you look at a picture of the brain like this, you can see the gray matter on the outside. That's what we refer to when we talk about ourselves, because that's really who you are right there. Okay? All your thoughts and stuff are happening in that gray matter. Of course, it needs to be able to communicate with each other, so all the white matter is there to make sure that all of your thoughts are organized. Okay. So as we see, white matter primarily consists of myelinated axons of many neurons. Gray matter would be neuron cell bodies, dendrites, axon terminals, unmyelinated axons, and all the neuroglia. In the spinal cord, we see the white matter surrounds the inner gray matter, and in the brain, the gray matter surrounds the white matter. Okay. So then I was going to introduce the nerve impulse. Gray matter is definitely working while you're studying, you betcha. Connections are being made all over the place, Suki. For sure, for sure, for sure. Okay, so when you go through an action potential, so that's what this is showing here too. Did I not have the picture? Yes. Okay, so right now you may be holding a pen to take notes, right? So you have a sensory receptor there, which we know is the dendrite of a unipolar neuron. Okay, so it'll be specialized for touch to hold your pen. Okay, that will send an impulse into the spinal cord. And you can see here there's the cell body of that unipolar neuron in the dorsal root ganglia. Okay, that's going to synapse and travel up to your brain, specifically to your thalamus, because everything goes there first. And then it goes up to your brain and tells your cortex that you are holding your pen. but you want to move your pen. You're not just holding it, you picked it up to write with it. Okay, so now the motor part of your brain is going to send an impulse down. It's going to synapse with a multipolar neuron. It's going to release a neurotransmitter, and that is going to then go down to the muscles that move your finger and cause a muscle action potential and move your fingers for you to be able to write. And that's what this is. The sensory receptor has something called a graded potential. An action potential we're going to see is all or nothing. Once it begins, it happens. A graded potential is not. They build up and then hopefully cause an action potential. So graded potentials build up. Once that, remember I said about we were talking about, and Crystal said the dominoes, right? Or was it Angela? Anyway, somebody said about the dominoes. Okay, so once you knock down the first domino, that would be an action potential because all the dominoes will fall. But building up enough breath or energy to be able to knock that first one down would be the graded potential. Okay, so a graded potential builds up in the dendrite portion, okay, and then hopefully builds up enough to trigger an action potential. When it does, then that travels. But every single time it has to make a synapse, you have another graded potential. If the graded potential is enough, it stimulates an action potential. But then you have to have another synapse, so that will be a graded. Okay, as we have learned, all of your perception occurs in your cerebral cortex. Okay, so that's that outer gray matter. That's where all your perception occurs. To be aware of anything, it has to make it there. As I mentioned to you, there's this little area here called the thalamus where all your sensory information goes. But it doesn't always send everything on. Okay, so, and you wouldn't have conscious awareness of it there. Conscious awareness is perception, so that has to get to your cortex. Okay, and then you're going to have a motor response. So again, every time you have a synapse, it would be a graded potential. If it's enough to knock down that first domino, you have an action potential. Okay, once that synapse occurs at a neuromuscular junction, it's a muscle action potential, and your muscles will contract. Okay, so that's what we're going to be looking at right now is the graded potential and the action potential. Okay, was that a nice review of last class? All right, so after this class, or it might have already popped up on Wiley Plus, you have quiz number 4, and it is on the PowerPoint we just finished on neurons, myelination, and neuroglia. Okay, there's 10 questions timed for 10 minutes. Remember those little quizzes we do once in a while? Okay, and that'll be due, I should know this, I think it's due by the next seminar. I'll confirm that on Friday night with you guys. Okay, so quiz number 4 is up. Thank you, Crystal. It's due March 28th. Okay, quiz number 4, 10 questions only, and timed for 10 minutes, and it's on the PowerPoint we just reviewed. So neurons, myelin, and neuroglia. Okay? This stuff that we're about to do is not on the quiz. Okay? Nothing about impulses yet on the quiz. You'll be getting that next. Because I love ya. Okay. So I asked you to look over pages 413 to 418 for tonight, and I'm sure you were looking at it going like, what, do I have to do the wiring in a house or what? Never mind, one of my friends works for Hydro, and like she's an uppity up and the safety officer and stuff, and she had to come to my house one time because my son had wired a lamp in the basement, and the power kept cutting out to it and stuff. So she was coming by anyway. So she says, well, I'll take a look at it. She had to rewire it all because he was going to start a fire. And we were lucky it hadn't happened yet. And anyways, electricity is complicated, and you really shouldn't do stuff like that if you don't know what you're doing. And boy, did she lecture my son. It was awesome. She's like, safety, you got to do this, and you got to do this, and you can't cross like that, and that's too close to that. And it was awesome. I was really proud of her. Anyway, in neurons, the nice thing is they're electrochemical, right? So electrical signals in neurons are electrically excitable because of that cell membrane. If you guys recall all the way back to chapter 3, we had learned about gradients being created by cells. Okay, so electrically excitable gradients would be a voltage difference. And as we just talked about, a voltage difference would be created by ions. Okay, there's two types of electrical signals, which we just saw in that last PowerPoint. Action potentials, which are the all or nothing. They travel the long distances down the axon. Graded potentials are very local, and they have to build up to knock down that first domino. Okay, so in a living cell, we are electrochemical. We can't just be electrical. Oh, the Wiley quiz number 4 is due April 4th. All right. I will confirm which it is on Friday night. I'm writing myself a note right now. Friday, confirm, due date. I'm sorry, I know I should know it, but I will make sure it's the same in every place, okay? Either way, you got time, so chill. How's that? You don't have to do it tonight. All right, so everybody's April 4th. I'm just going to double check, but okay. All right, so ion channels. Okay, so if you look on page 415, figure 15.9. Okay, on Friday night, we're going to label this picture, by the way. I've got it for you. There are different types of integral proteins in a cell membrane. Do you remember that when we drew the cell? And I had us draw leakage channels and gated channels and open channels. Do you guys remember that? Or leakage, gated channels and the pumps. Do you remember drawing those into the cell in chapter 3? Yes. Okay. Well, I made you do that way back then, so you started to understand a cell membrane because of the neuron. So if you still have your diagram from back then, oh yeah, Crystal, that's right, you weren't there. Okay, but if you remember that from back then and you still have that big drawing, you should go back and look at that so you recall that we did draw in those 3 different types of protein channels. Okay, so the leakage, ones that don't have gates, okay, also called open channels. They're always open. Okay, but they are still specific. Okay, so if we look at a nerve cell membrane, you would have more potassium leakage channels than sodium leakage channels. In other words, sodium's always leaking into your cell, and potassium's always leaking out of your cell through these specific channels for them that are always open. But there's more potassium than sodium. Everybody listening right now, it's 50 times more potassium channels than sodium channels. So you're going to have 50 leakage channels for potassium to 1 leakage channel for sodium. That's important. Okay? So in the picture on page 415, if you're looking at it, which I actually think I have it right here as well, okay, it shows 2. Okay, leakage channels for potassium to 1, but it's not 2 to 1, it's 50 to 1. Okay, so that's important. Like there's a lot more potassium leaking out of a cell than sodium leaking into a cell. Okay, did everybody hear that? That's really important when you're looking at the differences in charges. Okay, so because they're both positively charged, but you've got 50 to 1 of positive leaking out to positive leaking in. 50 potassium leave to 1 sodium coming in. Get it? Okay, so that's important. Okay, so there are way more potassium leakage channels than sodium leakage channels. As a result, they say that the cell membrane is more permeable to potassium. Obviously, a lot more potassium is leaking through it. That helps explain what we're about to look at. The resting membrane potential of minus 70 millivolts. Okay, we're about to look at that. But think about what that name means. At rest, the membrane has a potential. Okay, that potential is that it can eventually have an action potential, not just be resting. Okay, so you got to set that up somehow. So you set that up. If you're a living cell, how else are you going to do that? You're going to make sure that you're controlling the doors. Just like you want to control who is in your building and out of your building, you have doors to control that. Okay, big concert, only let them in certain doors, out certain doors. You don't want them coming in all over the place because they got to pay to get in. You got to make sure people are out. You control the entryway and the exits, right? Entrances and exits. That's the way the cell does it to be able to have a nerve impulse. Okay, you're also going to have, and don't ask me why they named them this, but they're called ligand-gated channels. Does anybody know what ligand stands for? Okay, it would be so much easier if they just called them chemically gated channels. Okay, so ligand equals chemical. Okay, molecules, yeah, but they're basically chemicals. Okay, so that's your neurotransmitters. Okay, so you're going to have neurotransmitters that can open or close these channels. Okay, I just wish they'd keep it simple. Okay, so voltage gated, that one makes more sense. Okay, they change to a direct change in the membrane potential. So they're all about the positively charged or negatively charged ions. Okay, if there's enough of those going through, like I told you, 20,000 sodium, when you have an action potential, that changes the voltage. So then a gate further down, when that voltage changes like that, it pops it open. Okay, so you have voltage-gated channels. As we learned, voltage-gated channels are all along the axon. Okay, and then we have ones that are mechanically gated. These are your mechanoreceptors, and you guys have learned about those. So your Pacinian corpuscles and your Meissner corpuscles and your type 1 and 2 mechanoreceptors. All of those are mechanically gated or mechanoreceptors. So they respond to a vibration, a pressure, a touch. In other words, they're forced open. Okay, so those are the different ion channels. In this PowerPoint, I just like to remind you when we did take some of the different channels that are in a cell membrane. Okay, so if you're not sure about some of these different channels, go back to Chapter 3, page 50, find your diagram that we drew if you still kept it, and you'll learn about all the different proteins that are in the integral proteins that are in the cell membrane. And we're just looking at the open or leakage channels, the chemically gated channels, the voltage gated channels, and the mechanically gated channels. Okay, when we're talking about neurons. But we have lots of different proteins, if you recall, in the cell membrane. So to create a resting membrane potential, the cell has to be able to control these ions as they cross. Okay, so we just saw that there are many more leakage channels for potassium than there is for sodium. So if we counted it like for every channel only one goes through, you'd have 50 potassium leaking out to 1 sodium leaking in. So you can see that makes a difference right there, right? But then if you look over here, we also have a pump, the sodium-potassium pump. And for every 2 potassium that it puts back in, it puts out 3 sodium. It's 3 to 2 on the sodium-potassium pump. So you're getting 50 potassium going out and 2 being pumped in by all the sodium-potassium pumps, and then 1 sodium coming in, but 3 sodium being pumped back out. And these are all over your cell membrane, the sodium-potassium pump, busy trying to keep this resting membrane potential. Okay, so this is why a high salt diet makes you tired, right? You have all these sodium, see the little green guys here in your extracellular fluid? When you increase that gradient, it means more sodium leaks through, and it's going to change the membrane potential, so your sodium-potassium pumps have to try and keep that gradient. Because if you remember, diffusion increases when you increase the concentration of the substance. Is this all reminding you of chemistry in chapter 2 and you were all so excited when you were done it? It's back! Anyway, but it's interesting chemistry. Okay? It's interesting chemistry. This is why you can have nerve impulses. Like, this is just super fascinating. Okay, so the cell, and it doesn't just have a few here. We know it has like 50 leakage channels for potassium to 1 sodium leakage channel. And of course, there's hundreds of thousands of these. And then it's going to have thousands of the sodium-potassium pumps. And all of those doing all that they do, the leaking out of the potassium, the leaking in of the sodium, okay? The sodium-potassium pump pumping out 3 sodium for every 2 potassium constantly. Of course, it's a pump, so it needs ATP to do that. It requires a lot of energy to keep pumping those back and forth, back and forth. It's not just a make work project, all this leaking and then pumping it back. It's not a make work, it is a make action potential, make a possibility for a nerve impulse to occur. Okay? It's what is basically life. It's why we're alive. It's why we have thoughts. It's why we can move, is because of these leakage channels and the sodium-potassium pump. Okay, now a couple of other things contribute to what ends up being this minus 70. You can see the negative all along the bottom here on the inside and the intracellular side of the cell membrane. We also have phosphates and proteins inside our cells that can't leave the cell. As you can see, they have negative charges. Okay, so they contribute toward the negative along the cell membrane on the inside. Okay, and then we have all this sodium outside, but we also have another anion on the outside, chloride. Okay, you do have chloride channels as well, but we'll talk about those later. Okay, because they don't contribute to the resting membrane potential. Okay, so all of this, these negative proteins and phosphates on the inside, all the potassium that's leaking out, the sodium-potassium pump putting everybody back, leads to this slight negative charge or difference between the inside and the outside, and they always refer to the inside as minus 70 millivolts. Now, just so you understand, that's the difference between the inside and the outside is minus 70 millivolts. Okay, so it's not really like it's minus 70 here and plus here. It's the difference between the inside and the outside is minus 70. Okay. Ooh, potty training. Everybody's favorite. Not. And absolutely listen to it. And we are going to be talking about this a bit on Friday night too. Okay? So this is how you create the resting membrane potential. So what I want to do, what time are we at? Okay, I want you guys just to take a quick little micro break, go check on the kid on the potty, and do what you got to do, because I'm going to pull up an animation. We can't listen to it, but I'm going to talk about it. Why can't I see it now? Where are you? It's got to be that. Yeah. Oops. What did I just do? I'm messing with stuff. Okay. Let's do this. This. Log in. Okay, so guys, just if anybody's still there, if you run to the washroom, that's fair and fine. Ooh. No, that isn't normal. What the heck? Yeah, I don't know. All right, so we'll figure it out when you're at school on Friday. Okay. It's not this one I want. All right, so here we are looking at the cell membrane, right? And we can see that here's the little voltage meter, and they're showing here how it's helping to build up to that minus 70 on the inside compared to the outside. Okay, so all that is all of these things happening together that lead to it. So we looked at the sodium-potassium pump, the leakage channels for sodium and potassium, and then when you have an action potential, you're going to need those gated channels. Okay? So there are three different, why isn't it running? There we go. Three different types. Again, we have the open pores, the channels, the pumps, and the gated channels. So here you can see the open pores and sodium going in and potassium going out. And remember that those are 50 to 1. Okay? This is when you have a gated channel to be able to have an action potential. Okay, and then you have the pumps that help maintain the resting membrane potential, and they require energy. So you saw 3 sodium going out for every 2 potassium being put back. So when we look at the resting membrane potential, which we were just looking at, you can see here we have all this sodium in the outside. We have all these proteins and phosphates on the inside. And we end up with this negative along the inside and the positive along the outside. Okay, it doesn't mean that the cytosol or the extracellular fluid have those charges. They don't. Okay, it's just along the cell membrane. That's really important to understand. So it's just the movement of ions across this phospholipid bilayer that creates a slight difference in charge between the outside of the cell membrane, of the neuron cell membrane, to the inside of the cell membrane. Okay, so to help maintain that, you have those channels. Okay, so you're going to have, as you can see here, one leakage channel for sodium, and you're going to have 50 actually for potassium. All that potassium leaving, all that positive charge leaving, is helping maintain that negative on the inside. As we can see, the sodium-potassium pump using ATP is going to put out 3 sodium and put 2 potassium back in. Okay, the sum total of all of that, it kind of sounds like math, you end up with minus 70 millivolts difference from the inside to the outside. Okay, we're going to watch this in class together on Friday as well. Okay, but if you saw where I went, this was under the C, membrane potentials. Okay, so it starts off with the resting membrane potential, which we were just looking at. Okay, and then it builds into the action potential. Now, as we all know, I can't leave Wiley Plus open. So we'll go back here. Okay, so we were just looking then in the animation about these working. Okay. There's also a really good website, I'll pull this up for you, on the sodium-potassium pump. Okay, there's this really good article, I put it right in the PowerPoint, about how that works. If you're interested, this is a good little article. It's not that long, okay, but it discusses about the sodium-potassium pump, and it's pretty interesting actually, okay. So I threw that in the PowerPoint for you, okay. Okay, so that's how we create the resting membrane potential. Okay, it's just by those leakage channels that are open all the time, sodium leaking in a little bit, lots of potassium leaking out, and then we have the sodium-potassium pump maintaining that. Okay, so a graded potential will kind of play with that. So it's a small deviation from the minus 70. I like to think of graded potentials as Stewie talking to his mom on the Family Guy. You've probably heard it. It's the ring of my phone with my boys call me. Mom, 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 mom. And if it builds up enough, mom screams. That would be the action potential. So graded potentials are getting enough of a stimulus to cause an action potential. Or, as we see here, the hyperpolarization, which would actually be kind of, I'm trying to think of an analogy, but it makes the cell even more negative so that an action potential is harder to have. Okay, so when you do the mum, 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 mummy, 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 what? Depolarization is when you create the action potential. You've pissed mom off. Okay? If you want to make it more negative, if you don't want mom to have a reaction, you do your chores, you clean the house, you're quiet, you're a good kid. How's that for hyperpolarization? That just popped into my head. Okay, so you're trying not to have anything happen. So instead of having a resting membrane potential of minus 70, you actually make it more negative. You bring it down to about minus 90 as most hyperpolarizations. There's another term for that we use all the time when we talk about the gate control theory of pain. Does anybody know that word? When we make a nerve impulse hard to have. Inhibition. Good job. Yes. Okay. Hyperpolarization is really inhibition. Not exactly, but it is close enough. Okay. So inhibition is what we're talking about. When we inhibit something, we're going to make it harder for it to have an impulse. So when we talk about, like in the Golgi tendon reflex, and we say that glycine is released and it inhibits the motor neuron going to the muscle, and then therefore the muscle relaxes, right? We've used that term a lot in the tendon reflex. Glycine causes hyperpolarization. Okay? It makes the membrane more negative so that an impulse is harder to have. So you're not going to have as many impulses, and therefore the muscle relaxes. Okay, so graded potentials can do that. Okay, it can do a hyperpolarization, or it can bring it to a depolarization. It can make it more positive and bring it to having an action potential, which would be equated, stimulation can be equated to depolarization, whereas inhibition can be equated to hyperpolarization. Okay, so graded potentials can do either. It can make it harder to have an action potential, take it further away so it's harder, or it can bring it closer to an action potential happening. The cell will depolarize and we will have a nerve impulse. Okay, so graded potentials can be either. So think of inhibition as you're inhibiting in the Stewie thing. You know, Stewie's going to be doing his homework, he's going to be cleaning his room, he's going to be doing everything, just don't piss off mom. Okay? Whereas, mom, 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 that's going to lead to an action potential. Something's going to happen. Okay? So hyperpolarization equal to sort of like inhibition, depolarization, stimulation. And graded potentials are bringing you closer to either or, either turning it off a bit or bringing it to where it can be turned on. And they build up, they're graded, they vary in size. Okay? They are also local, and they happen most often in the dendrites and cell body of a neuron. Okay, so if we look at this picture, which you will find in your textbook on page 416, okay, this shows us what's happening with the gates and what happens with that minus 70. So we learned already that minus 70 is our resting membrane potential, right? So that's where the leakage channels and the sodium-potassium pump get it to that. Okay? So we have this minus 70 that the neuron's sitting at all the time. Graded potentials happen between there and the minus 55. Or if it's a hyperpolarization, it can go below minus 70. And you can see that here, the hyperpolarizing phase, it's going below minus 70. So that would be a hyperpolarization. So here we are at minus 70. We're a resting neuron. So mom's sitting in the living room. Okay? And then Stewie, mom, 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 mom. That's a graded potential. If he bugs her enough, it gets it to something called threshold. Okay? Okay, thanks for letting me know, Angela. Okay, so you get to minus 55. That's called threshold. Guess what? Guess what's at threshold? Do you remember when we were talking about the trigger zone and the very first domino? The first domino's at minus 55. So this is where the graded potential is built up enough to knock over the first domino. So really, the first domino, no, your body will find the salt, but then eventually, yes. Have you ever heard somebody be in a marathon and they run out of electrolytes and their heart stops? That's what happens. Okay, because your heart has action potentials too. That's why it contracts. Okay, so you need your electrolytes, and electrolytes are your sodium and your potassium and your calcium and all of those things you need to be able to have your neurons and muscles work. Okay, so when people when they sweat too much, when they exercise too much, when they are starving to death, things like that, they run out of electrolytes, and then that's issues because things stop working, like your heart. Yeah. Okay, so we have this graded potential. Yes, yeah, you do need some salt and electrolytes. Yeah. Okay, so mom, get to minus 55. You say it enough, knock over the first domino. Now this is when that first voltage gated channel opens up, and remember those are in the axon. Zoom. right here in the green section, that's your action potential, your depolarization. That's your nerve impulse right there. Sodium channels opened up. Okay, so they open up, 20,000 sodium cross the cell membrane. They have a charge, right? They're all positively charged. They bring the inside of the cell membrane up to about plus 30. Different places say different things. I think Hank in his thing says plus 40 maybe. Okay, but in your textbook it's plus 30. Okay, so it goes up to plus 30. And then immediately the cell is going to get ready to do this again. Because look, this is all in milliseconds, right? Okay, so immediately certain other channels open up to dump out some of those positives. It's actually potassium channels because we have more of those. So let's open up those, dump out some potassium to start getting it back to resting so we can have another impulse. Okay, then once it gets back to the resting membrane potential, it can have another impulse. The time when it can't have another impulse, and you can see it over here, the absolute refractory period, is when the certain gates are open and you can't open an open door. Okay, so if the door is still open, you can't open it again. So you can't have another impulse until the door closes. Does that kind of make sense? You can't open an open door. So you have to wait till it closes before you can open it again. Okay, so that's basically what a refractory period is. Okay, so you're going to have a absolute refractory period here when the sodium channels are still open because, like I said, you can't open an open door. It's already open. And a nerve impulse is opening up the sodium channels. So refractory period is when you can't have another impulse until those doors are closed. Okay, so refractory, can't have an impulse. Okay, you have a relative refractory period when everybody's kind of going back to where they need to go. With a really strong stimulus, you probably could have another impulse. Okay, that's a relative refractory. But absolute refractory is when the door is open. Can't open an open door. Okay, you can't open it again. Okay, so depolarization, which is stimulation. You'll have a chemical or mechanical stimulus cause that graded potential, and it needs to get to threshold, minus 55. At minus 55, that's where in that trigger zone, okay, in the axon hillock, you'll have that first gated channel, and it pops open, and sodium rushes in. Okay, when sodium rushes in, 20,000 sodium enter the cell, okay, and they change the membrane potential up to 30. Okay, that's what your action potential is. Remember I told you that a long time ago. A nerve impulse is just sodium entering your cell. Okay, now there's two gates that happen in a sodium channel, and I don't think, yeah, I don't have the picture here. I have the picture already copied for you for this weekend, okay, where you're going to see the two gates. Okay, but in the sodium channel, there's actually two gates. There's an activation gate, which opens up when you stimulate it, and an inactivation gate that closes right after you do that. Okay, and don't worry, you're going to get a picture of this, and we're going to do, we're all going to get up and move and do this, so you'll get it. Okay, so the activation gate is what opens when the sodium go in. Okay, so it opens up at minus 55. The sodium rushes in. Almost right away, the inactivation gate stops it. So 20,000 sodium rush through, and then this other gate closes it off. Okay, so that's the depolarization. That first gate opens up, 20,000 sodium go in. We have now had our impulse. The inactivation gate stops that from letting more in, and that's it. Now, There's those gates at every neurofibril node in a myelinated axon or all along the cell membrane in an unmyelinated one. And they're all going to open up in response to that 20,000 sodium. So all of those gates are popping open all the way down the axon. But back at that first one where the inactivation gate had closed, okay, then the voltage gated potassium channels open up to try and get it back to normal. Yes, okay, so we can see the voltage gated voltage gated sodium channel activated, I can't speak anymore, sorry, my mouth just quit working. The voltage gated sodium channel activation gates open when we get to minus 55, okay? So that's what's happening here. They're all opening, okay? So then at the top of the curve, you're right, the inactivation gates stop that from happening now. No more sodium can rush in. Close that second door, stop them from rushing in, okay? Now we're going to open up voltage-gated potassium channels to try and get it back to normal so we can have another impulse. So now we have potassium channels opening up. Okay, so potassium channels opening. They're a little bit slower than the sodium channels. Okay, but when they do open, because they kind of happen at the same time, but potassium's slower. Okay, so potassium outflow starts and it's going to return the membrane potential back to resting at minus 70. However, a lot of time more potassium goes out, it goes to about minus 90, and then between the sodium potassium pump and everything else, everything eventually goes back to that minus 70. Okay, so again, going back to that picture, we have graded potential. Okay, so you're going to have a neurotransmitter, or you're going to have touch or pressure or something, push, 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 push. Pop open that first voltage gated channel at threshold minus 55. Sodium activation gates open, sodium rushes in. Once about 20,000 sodium cross the cell membrane, the inactivation gate closes and stops that. Okay? Correct, Liz. Yeah. Okay? Closes that off, then the potassium gates open up and we start getting more potassium out to get it back to normal, because it's got a positive charge too, just like the sodium, right? So we're talking about charges right now. So potassium is rushing out to get it back to minus 70. Sometimes it overdoes it a bit, so we get that little bit of hyperpolarization between the sodium potassium pump, etc. It gets it back to resting at minus 70. Okay, so that's repolarization, getting it back to being able to have an impulse. And just so you understand, at some point science has been able to study all of that, what gate opens when, what ion is moving when, and all of this is happening in milliseconds. Wrap your head around that for a second. Okay? As we saw, the refractory period is when it can't have another impulse. Okay? So it's in 0.4 milliseconds in a large nerve fiber. So in your A fibers, that's 0.4 milliseconds that it has an absolute refractory period. Okay, and that's when you can't open an open door. Okay, the inactivation gate is closed, the activation gate is open in the sodium channels, and so sodium couldn't go in anyway. Okay, the relative refractory period is everything's kind of going back, but hasn't gone back to its full resting state. So you could have an impulse, but it's going to take a lot. Okay, it's going to have to kind of force it. The door is kind of creaky still. Okay, so absolute, you can't, like I said, open an open door. All right, does that kind of, in any way, shape, or form, make somewhat sense to you guys? How about anybody else? Crystal starting to, Liz, Natalie, kind of, Kate. Leilani just needs some time. Yeah. Maria, yes. Natalia, kinda, yes. Okay, Elena. Brian, you there? How are you doing with it? Some time, Courtney? Yeah, and we're going to play with it some more, right? We're going to go over it next month. Okay, keep trying. Don't give up. Kind of. Okay, because a lot of the stuff we've learned to hear, right, is the synapses and stuff that we see in the tendon reflex. And when we look at, yes, Alexia, it will be going over it, and we are going to go over it again on Friday night, but this time I'm going to get up, and some of you are going to be sodium, and some of you are going to be potassium, and we're going to be running in and out of doors, and it's going to be so much fun, and it'll start to make sense. But if you, as Maria and I were talking about earlier, if you start to understand, we've talked about edema, and we have talked about electrolytes, and we have talked about the importance of not having too much sodium in your diet and having electrolytes, and it all comes down to this. This picture right here, this depolarization and stuff. This is life right here, okay? I sound like Frankenstein. It's alive, right? But it is. This is life. So the more we can understand this, the more a lot of things will make sense when we talk about neuropathology and what happens to people when their nervous system doesn't work very good, or even just when you're super tired because you had McDonald's fries. Like, it all starts to make sense the more you can understand these, okay? So before we move in into continuous versus, can we take a little bit of a micro break? Recording again. Okay, so let's take a look at the animation. So when we talked about the graded potential in the PowerPoint here, that's the spot between -70 and -55, right? So graded potentials happen in that little blue section there, going from -70 to -55. When we look in the animation, we can see that those happen in our unipolar neurons and our sensory neurons, where we have the receptors, right, the dendrite portion, or the other nerves that happens in the dendrite portion right by the cell body. Okay, so like in a multipolar neuron, that's where the graded potential. So what it wants to do is a graded potential wants to build up enough to affect that trigger zone and knock down that first voltage gated one, right? And so that's going to happen before the one long process in the unipolar neuron, or at the axon hillock here in the multipolar neuron. Okay, so let's just take a look at that happening. So there's our graded potential. That's the mommy, right? So what's happening is you're trying to get it from -60 to get up to -55, because we know that that is threshold. If it gets to there, we're going to have enough to be able to have a depolarization. But if we want to make mom happy, right, we do our chores and stuff, we actually want to make it more negative, a hyperpolarization, making it harder to have an impulse, because then it's going to have to go from this -90 or -100 to get to that threshold of -55. So this one, the bottom one in this picture, hyperpolarization, is when what we would call inhibition. Okay, when we're inhibiting something. When we're stimulating something, we're going to get it to -55 in threshold, and an action potential will happen. In a hyperpolarization here, you saw that it was opening up more potassium channels. Let out more positive, make it more negative. Okay, you have another option with that, though. It depends which neurotransmitter is being used. But if you remember, when we were looking here at the resting membrane potential, there was these negatively charged chloride ions, anions, in the extracellular fluid. If you wanted to make the inside more negative, could you not open up channels for those and let those through and make it more negative? Yes. Okay, so different inhibitory neurotransmitters do it different ways. Some will open up the gated channels for potassium and let even more potassium out, which we know is what contributed to it being minus 70 on the inside. Or you could just simply open up channels for chloride, let it in, it's negatively charged, and it will make it negative on the inside. Okay, so you have two ways to hyperpolarize. Okay, and we know it's sodium gets let in to be able to get it to that minus 55. So that's the graded potential. Okay, so that's getting it to the minus 55. So here you can see it can either increase or decrease. I'm just going to skip that. Okay, well, actually I won't. Okay. So here we can see it's going to either go more negative, okay, hyperpolarizing graded potential, or it's going to try and get it to threshold and be making it closer to that threshold. Okay, hyperpolarizing, making it more negative by either opening up chloride channels or letting more potassium out, or it can let more sodium in and push it closer to threshold. And we know once it gets to threshold, we're going to have an action potential. Okay, so we can see in this picture, action potentials happen down the axon. They're called all or nothing because once that first voltage gated channel opens, or the domino effect starts, once that first one opens up, the rest are going to open up because all along the axon are voltage gated channels. Okay, so as soon as that trigger zone gets stimulated, you get that first voltage gated channel opens up, they're all going to open because the 20,000 sodium stimulate that. Okay, so here you can see the sodium gate actually has two gates. So an action potential, once the graded potential gets it there, okay, this is the green line in this picture is the action potential. So the first thing that has to happen is we have to let sodium in. Everybody agree? Okay, so look at the sodium channel here, the gated channel for sodium. A graded potential occurs. Here's our activation and inactivation gates. Okay, we have the two gates on sodium. The graded potential will occur. Okay, and it's going to be enough to pop open the activation gate once it gets there. So the graded potential, trying to get it to minus 55 to pop open that gate, because that's the only thing that's going to open up that gate. There it's opened. 20,000 sodium come rushing through. Okay, the inactivation gate is triggered by, once there's enough gone through, they're activated by the voltage as well. Okay, so activation gate is open right now, so is the inactivation gate. But as soon as enough sodium goes through, the inactivation gate stops it. At the same time, the potassium gate opened up. Did you see that? And it starts bringing it back to normal. Okay, right now, could sodium have entered with the inactivation gate closed and the activation gate was open? Could sodium have gone in? No. That would be absolute refractory. Okay, so you have the two gates in the animation, not in your textbook, but I have the picture for you on Friday. I have a handout for you that has that. Okay, so this is the picture we're just looking at anyway, so I'm just going to get rid of this now. We'll go back to the PowerPoint. Okay, so again, we have a resting membrane potential of minus 70 created by leakage channels and the sodium-potassium pump. A graded potential will occur, whether that's touch, pressure, or trying to stimulate your motor neuron to contract your muscle. A graded potential occurs. If it's enough, it pushes that cell membrane to about minus 55. Minus 55 is called threshold. That's when the first voltage gated channel opens up, or the activation gate in the sodium channel opens up. Once that opens up, 20,000 sodium, zoop, go through. Once they go through, that triggers that inactivation gate to stop that. So that stops them from coming in. Just as quickly, the potassium channel, gated channels open up, and potassium starts flooding out just to get it back to where it was so it can have another one, another impulse. Okay? Then everybody eventually gets back to normal, and we have another resting membrane potential. Okay? And we can have another impulse. And all of this is happening in milliseconds. Okay? Now, we looked earlier at myelin. So continuous conduction is what happens on unmyelinated fibers, those C fibers, those really slow ones. Okay, so if we look at the picture here, the current or the sodium voltage gated channels have to open all along the axon. Whereas when you have saltatory conduction, which is a myelinated fiber, saltatory, it's jumping, it's dancing, it only has to open up channels in the nodes of Ranvier or the neurofibril nodes. So the current flow tends to skip along, and that speeds things up greatly. If you recall, in continuous conduction, this would be in your C fibers. If you go to page, where is it? 418, and it shows the effective axon diameter there. In a C fiber, which would be this continuous conduction because it's not myelinated, you see that those fibers conduct an impulse at about 2 meters per second. Whereas if you look at the A fibers, okay, they're at about 130 meters per second. So myelin greatly speeds up an impulse, 2 meters per second versus 130 meters per second. Okay, so it obviously takes a lot more energy for continuous conduction because you're going to have to, along this entire axon, put all that sodium back, and everything has to go back, and those sodium-potassium pumps are working, whereas in a myelinated fiber, it's also energy conserving because you only have to do that at the neurofibril nodes where it occurs. Again, when you look at MS, another reason why it's so fatiguing. Okay. Okay. So the things that affect the speed then would be that myelin, the size of the axon. So again, you can see that on page 418, and we've already talked about that quite a bit, right? The A, B, and C fibers. And of course, temperature. When you're warm, your impulses travel much easier than when you're cold, which is why we use ice to actually help people with pain, because it actually slows down the impulse. Or right now, if you were to go outside and stand in the wind, in no time at all, you wouldn't be able to feel your face. I just saw quickly while we were taking the micro break, Highway 1 is closed to Ontario now. It's just beautiful outside. Yay, March, in like a lion, means it's going out like a lamb. Yay. Okay, but here's the important part about the things that affect the speed. It's not related to the stimulus. Okay, so in other words, an action potential is an action potential. It's all or nothing. Okay, it doesn't mean you're going to feel anything more or less. You're either feeling it or you're not. Okay, so the larger fibers just conduct it faster due to the size of it and the myelination. Okay, but the impulse is just the impulse. Like that is the opening of sodium channels. And once that first voltage gated channel opens up in the trigger zone, they all open all along the axon. Okay, so we see the A fibers, they're the largest, they're myelinated, and as we know, they get grouped because most of the fibers in our body are A by speed using the Greek alphabet, alpha, beta, gamma, delta. Okay, B fibers are medium size, and they are all about our autonomic nervous system, and that's all you need to know at this point. Okay, they're actually the preganglionic. But again, you don't need to know that. You just need to know that they're ANS, autonomic nervous system. And the C fibers, we learned were our slow pain, right? So they're unmyelinated and quite pokey slow compared to the other guys. Okay. All right, I think you guys have enough information in your head right now. What do you think? Y'all got real quiet, so I know you're done. Do you like this? Studying for finals. When did you become an expert in thermonuclear astrophysics? Last night. That's not the way you should study for tests, by the way. Okay. He can get away with it, but, you know, he's genius, and he's like a character in a movie and a comic strip. Okay. So on Friday, oh God, no. No,